Hey gang, say I'm gonna try and do a drawing today um, that'll help you understand this notion of going from a gene to a protein. And we're kind of introducing this thing called RNA, and there are three RNA players. But if we can kind of go through this drawing, I hope this drawing will help the textbook and the additional layering of this concept make sense. So we can see this round circle here. This is supposed to be the cell membrane, and this other inside fairly large circle inside of that would be the nucleus. And I can even label some of these things as we go along. So we have a nucleus, and then inside the nucleus, we have this twisted strand labeled DNA. So this should all be basically familiar from our investigations earlier on about the cell parts. So we got the nucleus. So let's assume, I like to think of RNA and the R for RNA meaning repair, or to rebuild, or to make something. So it kind of works like this. The DNA never leaves the nucleus, at least in a eukaryotic cell. And so if you need something, if the cell perceives it needs something, what we have to go in is we have to go inside the nucleus, open up a small piece of the DNA, get a copy, almost like taking a photocopy of that piece of DNA, and then we leave the nucleus, and the substrate that we use is RNA. So we leave the nucleus with our RNA, go to a building site, which is a ribosome, and we take that information and we use amino acids to put them together in the right order to create a final product, a protein, sometimes called a polypeptide, based on the information that we got from the DNA. So what's really miraculous is we have 30 some odd thousand genes on that DNA molecule. And when there's a cell need, that information is sent inside the cell so that we open up just the part of the DNA molecule that we need to read in order to get a particular protein. So let's take this example. How about you just had a bunch of milk and a whole bunch of ice cream, and now your body is loaded with lactose sugar from those milk products? So let's say that this cell needs to make a bunch of lactase, which is the enzyme which breaks lactose sugar, which is a two-part sugar. It's a disaccharide. It needs to be broken in half so those sugars can be burned in your body. So as a disaccharide, it's not very functional, not helpful for us. In fact, some of us aren't able to make this lactase enzyme, and so we're called lactose intolerant. So anyway, for the sake of demonstration, let's say that the information is found right along here, this from here to here. That's the information on the DNA molecule that codes for, here's how you make lactase enzyme to break apart lactose sugar. So the first thing that happens is we enter the nucleus, and so we're going to build this strand, and I'm going to draw it as if it's a continuous process. So I'll put a little arrow here. You know, it's kind of going this way. And we're going to take that strand of information, and when I say strand of information, that RNA, it follows the complementary base structure of the DNA. So for instance, across from A is T and C is G, normally in the DNA molecule, but there's one little rule here with all the RNA products, and that is that they're unable to make, or they cannot make T or thymine, so they make U or uracil instead. So across from A will be uracil, or U. Across from T will still be A. So anyway, more on that to come. But generally speaking, so we have this spaghetti noodle, if you will, this combined list of letters built upon this DNA information. It leaves the nucleus, goes to the ribosome, and this is called, this strand that was directly built using the DNA molecule is called messenger RNA, mRNA. So that's the first of three RNA players in this whole thing. But just keep in mind, the whole idea is to get some information from the nucleus and then build a protein. That's generally where we're headed. So now this fiber comes into this big blob sitting on the ribosome. We said the ribosome was a workbench of the cell. So that makes sense. We're going to build things on the ribosome. So this little blob is known as a messenger, or excuse me, the ribosomal RNA, which makes sense. It's sitting on the ribosome. So ribosomal RNA, or rRNA. Now we've got the information, and they're the word that we use in that getting the code from the DNA and making its sister code on the messenger RNA. That code creating, we're kind of transcribing. And so that I have this word transcription. That's this process right here going on. This reading and building, so the reading aspect is called transcription. Later on, the building aspect down here, we're going to call that translation. And there are two words that sound kind of similar, but the good news is they're alphabetical. In other words, transcription happens before translation. So easy way to remember that. All right, so we have this information, and it's going to occur in snippets of 3, and I'll lecture about that later, but essentially here's the deal. We have 20 different amino acids. I've drawn just little circles over here. Those put together make proteins. Those are the raw materials. There are only 20 different kinds, so your entire body is made from 20 different kinds, just like all the plants and fungus and everything else. 20 different amino acids. What's critical is we put them in the right order, and the code is read 3 at a time. So every one of these amino acids has a 3-letter code. And so if it was just one, we have 4 bases on the messenger RNA molecule available. We still have A's, we have C's, we have G's, and we have U's. So we have 4 letters. How do you code for 20 amino acids if you only got 4 letters? Well, we could do combinations like in pairs maybe. Like A and A would be one of them, and then A and C would be another, and then A and G would be another, and A and U would be a 4. Well, you know what? That gives you a total of 16, and that's not enough. If we read them 3 at a time, that is to say that if we have 4 letters to work with and we put combinations of 3 of those 4 letters together, our total combination is something like 64. So the way this works is for each of these 20 different amino acids, several codes call for the same amino acid. So there's redundancy that way, but we get all 20 covered with 64 choices. So anyway, there's a word for that. So I'm going to make a little hash mark. So let's say those are 3 letters and these are 3 letters. I'm going to put a space in there just for simplicity's sake. That space really doesn't exist. They'd all be equally spaced, the nucleotides. And so let's say I'm going to keep these simple too. So let's say this is A, and this is C. I'm just making this up. Let's say this is CGC, okay? That is the messenger RNA. It got this series of codes. I can write some more here too for that matter. Let's just do it again. CGC and AAA. So this information is coming from the complementary bases on the DNA. All right, so what we need to do though, we want to build something with these using this code. And so, I was going to mention, there's a word for every 3 that we have here. We call that a codon. A codon. All right, so read 3 at a time. It's really important that order that we start reading and we stop, and that the order is preserved in triplicates. Because a shift in that would be devastating. You put together the wrong amino acids, or maybe by shifting, we call it a frame shift. That would be disastrous. And that's why I've kind of separated these for easy viewing purposes. So the next piece that's out there are things called transfer RNAs. I think of them as little pitchforks, kind of like this. And there are some of them that are going to fetch more amino acids. So these are amino acids, and there are 20. I just threw a bunch of them up there. And so I have this accumulation, and so they have these little pitchforks called tRNA, which is short for transfer RNA. And they're just going to transfer, they're going to carry the amino acids to the proper spot on this workbench. And by putting the right cluster of these together, we'll have a protein. So let me add a few more out here so we can kind of see the whole process is going this way. And I'm going to go C, 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 and then G, 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 just for super simplicity. So what happens is a transfer RNA like this, if the code is the opposite, if we flip this code, so it says CGC, so the anticodon would be GCG, and this guy would come on over, and he'd carry his amino acid with him, and he would hook up just like this. So the anticodon is next to the codon. And so I'll even use the same color for my anticodon. So this is GCG. Hooks up. And prior to that, already hooked up, I'm going to draw this kind of in the wrong order, but already hooked up would have been the transfer RNA. And this one, let's say this one already hooked up, and now it's breaking free. And we have, so these two amino acids were brought to this building strand that we're making, this messenger RNA strand. And this would have been GGG. And earlier on, this was CCC. So we have the codon from the messenger RNA, and the transfer RNA is going to hook up. And once these are put in order, we start making these bonds that hold the amino acids together. And they get a fancy name. It's called a peptide bond. And up here we've got another one ready to come down to hook up. And so this is kind of going this way. And so let's see, this one's getting ready to come in to be locked